Hello, everyone. My name is Lewis, and my group mates are Marc and Jonathan. We will be presenting on the journal article titled Cyclin Synthesis Drives the Early Embryonic Cell Cycle, specifically focusing on figure 2b. Scientists are studying the role of cyclin in the process of mitosis during the cell cycle. They are studying this because it appears to develop numerously in interphase and then decline in number during the mitosis process. So it appears as if cyclin regulates the mitosis process and induces the maturing of oocytes. In this study, the scientists are using different amphibian egg extracts that have numerous cell cycles to show how cyclin is the only protein synthesized and required to induce the process of mitosis. Cyclins are important to study as their cycle of synthesis and degradation is integral in the cell cycle. These could be altered for different adventitious purposes or to avoid certain malicious diseases from occurring and multiplying. Main techniques used are injecting frog xenopus egg extracts with SAM-E before incubation and then analyzing proteins on SDS polyacrylamide gels to track the MPF activity throughout the different cell cycle phases. Then to test the synthesis of cyclin's correlation to inducing mitosis, cyclin mRNA is added to cell cycle extracts whose mRNA had been destroyed. Then the mitosis process is tracked with reference to time taken to occur by using 35S methionine on polyacrylamide gels. The model organism used once again is xenopus frog's eggs. Cyclins have been identified in a number of different organisms and therefore thought to be a protein synthesized for mitosis induction. The cell cycle regulation process is necessary for cellular homeostasis, in which internal regulations restrict or allow the cycle of the cell to proceed after preparatory processes have already occurred. Cell transitions occur after specific cell cycle checkpoints that have internal and external cues, making sure that cell cycle events occur in the correct sequence, as well as other key events to take place. The special cell type that was used in this experiment was electrically activated xenopus eggs that have been electrically activated to be released from CSF-mediated metaphase arrest, then washed in a buffer solution similar to egg cytoplasm, crushed and centrifuged to fraction them so that the ATP regenerating system, as well as deep membranated sperm nuclei, could be added for observation. To test cyclin, they used extracts from advanced or activated eggs in which the mRNA had been destroyed by adding mRNAs as well as RNasin to inhibit it. These cells' role is to test cyclin amount added in correlation to interphase length. The cyclin protein family is what the study is centered around, finding out its effect on cell cycle timing and regulation. Cytoplasmic factors involved are the cyclin proteins and mRNAs whose functions occur during the embryo's early development. Proteins include CDC2 and P34CDC2. This method was used to determine the effects of cyclin on cell cycle proceedings because by removing the mRNA and then adding differing amounts of cyclin mRNA, it allowed for the tracking and observation of the exact amount of cyclin required. This was unique as cyclin was not the focus of a study in relation to the cell cycle previously. 35S methionine is an amino acid labeled with a blue dye, 35S, that contains sulfur and is used to build many molecules such as proteins. The dye is used to label different metabolic or cell activities. It is added to the final concentration after the experiment is concluded, diluted by a power of 10, and then run on 12.5% polyacrylamide gels to show the protein synthesis pattern. The results from this process will be MPF measured against time taken in minutes. By taking the frog egg extracts with removed mRNA and injecting differing amounts of cyclin mRNA into them, observations were able to be made determining the amount required to induce the mitosis process. Then by adding 35S methionine and finally applying the different samples to SDS polyacrylamide gels, the MPF activity was able to be tracked by time in minutes as it progresses or doesn't progress through the different cell cycle phases. A research question for this figure is, do synthesized proteins extracted from an in vitro cell cycle with 35S methionine show that cyclin content increases during the cell cycle stages to induce the process of mitosis? The general approach used is to take the samples that were taken at specific times when different stages were occurring and dilute them with a sample buffer, then run them on a 12.5% polyacrylamide gel to determine their nuclear morphology. This will give molecular weight markers in thousands and show cyclin bands for each increment. Increments of time for samples taken were every 10 minutes from start to 80 minutes total. The control for this experiment is a non-modified embryo that has undergone the cell cycle and had samples taken during time intervals so that these results can then be used for comparison with the gel plates of cyclin-modified embryos. The data expected for the control is an increase of cyclin for interphase until nuclear envelope breakdown, and then declining for telophase. This figure shows an audio radiograph portraying cyclin content during the different cell cycle phases for an in vitro extract that had 35S methionine added to it at the starting point. Samples were taken at every 10-minute intervals, diluting using a sample buffer, and then ran on a 12.5% polyacrylamide gel to obtain nuclear morphology results.
Thank you, Lewis. I'm Mark, and to build off of what Lewis was talking about, here are some more relevant research questions related to our figure. In regards to the whole study, does cyclin drive the early embryonic cell cycle? Now, in regards specifically to this figure, what can be said about the behavior of cyclin bands in vitro and in vivo? During which phase of mitosis is cyclin most active? What is the relationship between cyclin and nuclear breakdown? And do the two primary cyclin translation products undergo post-translational modification? And if so, how many bands do they yield? We expect the cyclin bands in vitro and to be identical to those in vivo, as well as the levels of maturation promoting factor MPF and histone H1 kinase activity to be closely correlated. We also expect during the first and second interphase for cyclin bands to increase, but only to drastically decline between the nuclear breakdown and the onset of telophase, and for the two primary cyclin translation products to undergo post-transitional modification. Next slide. All right, so these pictures are just some visual representations of what took place in order to conduct this experiment. The frog embryo picture reflects the xenos frog's eggs and how the experiment was conducted within the organism in vivo and outside the organism in vitro. And in the top left corner, the picture of the frog eating or expelling a DNA strand represents the removal of the mRNA and then adding differing amounts of cyclin and mRNA for tracking and observation amounts of cyclin for the experiment. The remaining pictures represent the flow/steps taken in order to conduct a gel electrophoresis experiment. In this case, preparing the 12.5% polyacrylamide gel to obtain a nuclear morphology results. And then loading the time interval samples taken from the control sample of a non-modified embryo that had undergone the cell cycle, and adding with 35S methionine into the wells. Then running the 10-minute time increments for samples for a total of 80 minutes, and comparing the gel plates of cyclin modified embryos under the ultraviolet light to finish it off. Next slide.
Thank you, Mark. So again, the figure is just an audio radiograph of the protein synthesis in an in vitro cell cycle. It was ran through a 12.5 polyacrylamide gel, and samples were taken at intervals of 10 minutes, where protein fragments was trailed by the addition of 36S methionine. On the right hand of the graph, the researchers marked the cyclin band concentration for the viewer to follow the distinctive steps of the cell cycle. The markers up top are the time increments from 0 to 70 minutes, and distinct events occurring in the cell cycle will mark with NBD and TELO. NBD stands for nuclear envelope breakdown, which refers to the release of the contents of the nucleus into the cytoplasm, which is activated by MPF. Telophase is the final phase of cell division, where chromosomes repel to opposite ends and the formation of two nuclei begins to occur. And on the left side of the figure, we have the molecular weight markers, which were used to track cyclin by fragment size. And they identified cyclin concentration by noting the patterns of accumulation and disappearance of these bands in relation to MPF activity during times of the cell cycle. Early moments of the cell cycle show light thin bands of cyclin protein accumulating. Between 10 and 20 minutes, we see a distinct shift to black bands of cyclin as the cycle nears NBD. Cyclin is at its peak at 20 minutes as it has reached the adequate concentration to spark MPF production and lead the cell into division. The nuclear envelope breaks down and most of the contents of the nucleus flow into the cytoplasm. 10 minutes later, cyclin protein's level starts to decrease as the cell cycle nears the onset of telophase. At 40 minutes, we see a faint gray band as the cell is undergoing division for the first time. Now, between 40 and 50 minutes, protein synthesis rate increases, as noted by the black band reappearing once again. As the cell goes through the second interphase, cyclin concentration is now rising to signal MPF to carry forward the process. 60 minutes, cyclin has reached its peak concentration again, which was needed to signal MPF to initiate the breaking down of the nuclear envelope. Lastly, between 60 and 70 minutes, the band intensity in the gel begins to decrease as nuclear breakdown is occurring, causing cyclin levels lower to a less active state. The cell is now undergoing division. Next slide. Overall, the findings indicate that in vitro cell culture can meet the protein requirement to be able to activate MPF and carry the cell into mitosis. The two cell cycles observed on the gel describe the wave-like nature of cyclin synthesis. Early cyclin synthesis shows the gradual rise of the protein through the cell cycle, only to peak at the nuclear envelope breakdown and start its decline again. This explains that a certain amount of cyclin is needed to accumulate to trigger the MPF and carry the surge of reactions, thus leading into telophase. Another view of cyclin's role is that moments of cyclin degradation in the cycle are correlated with the initiation of the telophase. As the samples in the gel depict, protein band intensity fades down after the time of nuclear envelope breakdown and continues dropping through telophase 20 to 30 minutes in. It shows that the loss of cyclin in that 30 to 40 minute marker range seems to progress the cycle into the second round of interphase. Now, during the first and second interphase, cyclin bands increase, but only to drastically decline between the nuclear breakdown and the onset of telophase. Now, although its appearance before nuclear breakdown is important to understanding its method in driving the cell cycle, its degradation after nuclear breakdown events unfolds, highlights the importance that it could be playing in the exiting of mitosis. Next slide. A critique about this paper would be that band information is not labeled on the gel. Other band information is not labeled on the gel or mentioned in the paper, and no description is given about the extra material. Secondly, unlike the first round of mitosis, the protein synthesis after the 70-minute mark wasn't shown, missing out on the visualization that cyclin oscillates with the cell cycle. A suggestion is that the authors used a more clear format to capture the gel data. It wasn't a big problem, but increasing the clarity would have shown a more clear depiction of the rise and fall of cyclin synthesis.